Hi everyone, welcome back. In this video, which I think will be a bit longer than my normal videos, in this video I am going to focus on spatial transcriptomics, and this would be interesting for those of you with data science skills, you have coding skills, and you're looking for a domain that interests you where you can put your skills to practice. And this can be one of the fields, so you get exposed to what spatial transcriptomics is and what single cell sequencing is, and I'm not going to go a lot in depth in terms of what the technology is. We are going to cover a little bit basics in terms of what it is and then jump into analysis using one of the data sets. And for those of you biologists who already are in this type of fields, you probably know how spatial transcriptomics, this type of field, actually has exploded, especially in the last 5 to 8 years, like especially since 2015. There's a lot of new techniques that are coming up. No matter what technique you run into, eventually it comes down to working with a tabular data and performing some series of analysis to extract insights out of this data. So this video is about going through the basics initially for those of you who are new to the field. I don't bore you with a lot of details. There is a lot of information on YouTube for you guys if you want to build on some of these, but I want to make sure everyone is on the same page when we get to the analysis part. So I'll go through a few slides and then let's jump into Colab Notebook. As usual, I'll share the code, find the link down under the description. And while you're there, you will find the subscribe button. If you're not a subscriber, go ahead and click on the subscribe. And of course, if you're feeling extra generous, click the thanks button, especially if you like these type of videos. Okay, let's jump in. We have a long journey to take here. And starting with the first one. So if you are a data scientist, like I already mentioned, you're looking for an application domain, then try to think about bioinformatics. And again, this gives a first or quick glance at what bioinformatics is from a data analysis point of view. Obviously, bioinformatics is all about extracting information from your biodata. So here you see I took an excerpt from Nature Methods, I guess. It says Method of the Year, Spatially Resolved Transcriptomics. This was from 2022. I should have, yeah, right there, 2020 actually. I should have done my homework and be a bit more prepared. But again, this is a long video, so buckle your seatbelts. And if you read this article, somewhere along the way, this executive from or a representative from one of these biotech companies, basically this summarizes the reason why I want you to consider bioinformatics. Here it says the company has more programmers than molecular biologists and engineers. Yeah, this doesn't mean they don't need molecular biologists or engineers, but if you have coding skills, this can be one of the areas where you should consider, to apply your skills. Okay, with that, I think you're sold. Now you will watch the rest of the video to see exactly what type of analysis goes into these, these type of applications. Now, first thing first, let's understand what spatial transcriptomics is. Like it's made up of two words, and transcriptomics is transcript and omics. So let's go ahead and start with omics. I mean, you have genomics, you have proteomics, right? So you probably heard omics quite a bit. And omics basically means a collection, collective characterization. So you're not just looking at like a one single cell, you're actually looking at a collection of cells or proteins or whatever the molecules and translate that into, okay, what the structure is and the function and dynamics of this organism. It can be a mouse, it can be a human, but it's basically omics refers to collective characterization. And transcript, this may be new to non-biologists. It's a RNA strand that's produced when a gene is transcribed. You have a gene and you have like this specific structure and you can transcribe that. Transcribe means what the English term transcribe is. You're transcribing from one language to the other, or you're transcribing from one source to the other. So that's what transcribe is in this case. And as you can see in the center, in the middle, the top, you see the DNA structure. In the middle, you see how RNA is transcribing, which means if you see to the T molecule, like you have an A connected. And it was even previously when it was in DNA. In fact, I should use my mouse here to say here T and A and T, C and G, these are all connected, right? So when you strip this and when you are transcribing it with RNA, the A, which was previously connected to this T, is still being connected. It's just that RNA doesn't have this T it has U instead of T. That's why wherever you see T, I'm not going to get into the biology aspect of it, but you can go ahead and read that and get your own understanding of this entire process of transcription and translation. But in summary, wherever there is a T, replace that with a U for whenever you're talking about RNA, but A is an A, G is a G, right? So this structure, when you look at this RNA, you know exactly where it came from because, you know, it's complementing that. Okay, now you take that and again, this is what transcription is. Now you study the RNA. Now when you study RNA, you understand exactly what's happening at this level. Okay, that's what transcript is. And now moving on to spatial, you all know what a spatial is. Spatial is basically you're assigning at a spatial level. So this structure, to understand the RNA, you can do it in bulk. You can take like a whole bunch of cells and do your RNA sequencing, and then you know exactly what RNA, what genes are present in the sample that you actually took. But then mapping it spatially gives you specific idea about, okay, this is exactly where some stuff is happening. So maybe when you're devising your therapy, you can target these specific cells and not the entire thing. Why actually give medicine that affects the entire brain if you can target a specific region? In a way, that is one of the applications of why you're doing spatial transcriptomics. Okay, now with that information, let's move on. And let's look at the basics of DNA. And again, I may bore some of the biologists for the next 1 minute when I go through this slide. So if you want, you can just move ahead. But again, the idea is to bring everyone to the same page. So the DNA sequence is same in all cells of a given type of organism, right? So that's what we call a genome. When we say a human genome, we know exactly what the DNA sequence is. In the genome, you have 20,000, 30,000 plus or so many genomes in humans, and same with the mice and everything, right? So DNA sequence is same. And DNA sequence of an organism's genome, the one that we just mentioned about, it contains instructions, right? I mean, it contains instructions for the development, you know, hey, this is a skin, you know, so go ahead and generate some skin and so on. Generate red blood cells if it's in a bone marrow. So the organism's genome contains the instructions to develop and function and maintain that specific organism. Again, mouse, humans, you name it. And for a specific organism, again, let's focus on mouse, then the DNA sequence is consistent across all the cells within that organism. Right? I mean, you go to crime scene investigation, you see DNA. This matched this person. How do they do that? Because the DNA sequence is generally consistent across different cells, no matter where the cells come from within that specific organism. Now, in this mouse body, every cell contains the same set of genes, meaning the same DNA sequence, right? So, but how do they differentiate from one cell to the other? Because the cells may have different genes turned on or off. When I say different genes, it's actually a combination of genes. You have a combination of genes in these DNA that are turned on or off, making them perform some specific functions. Again, the cells in the bone marrow, they have the genes that are responsible for generating red blood cells on. But in the skin cells, they're not on because you don't need to generate red blood cells in the skin, right? So that's the idea here. And different cell types, they have different gene expression profiles, the example I just gave you, or you can think of muscle contraction. If muscle contraction is required, like in the heart, for example, then these genes will be turned on. And in certain cells, like skin cells, those genes will be off because you don't need to do that function right there. And excuse me, the cellular diversity, meaning how different these cells are, and the specific function is assessed at a DNA level, right? Not at the cellular, at a DNA level, but is assessed at a protein level, not at the cellular or the DNA level, because each of these are turned on or off. So in an ideal world, you would be studying these proteins, which people actually do, but there is no commercially available, or it's not that easy to quantify thousands of proteins. Why do that if you can get similar information from studying RNA? That's exactly the reason why there is the transcriptomics, actually the field exists. And in fact, in 2009, there was this first paper on mRNA, where the entire mRNA, 20,000 genes or so from a single cell, this is what we call it, or this is what's known as a transcriptome. This kind of literally opened the doors for this entire field of spatial transcriptomics. That's a good paper. Again, I'm flashing it on the screen. I'll try to provide all the links down in the description for you to easily find these, but very good read. Even if you are not a biologist, some of the terms may be a bit challenging, but you start uncovering what these terms actually mean. It makes a lot of sense, reading these papers. They're easy to read, I should say. Now, why study RNA? We already established the case that, hey, we ideally would like to understand information down to a protein level, not really at a DNA level. I mean, of course, that's also important, but really information at protein level is even more useful. Studying RNA can provide insights into these genes that are actually turned on. Again, when you read papers, you will encounter different terminology. I'm calling them turned on or off, but sometimes you read them as, hey, these are the expressed genes, or you'll hear the term gene expression. So RNA molecules are synthesized from the DNA template. So studying RNA will give you the information about this DNA, and RNA in a cell can provide information on what genes are actively being transcribed, because this is really important in understanding the biological function of what's happening down at that level. And of course, it allows the researchers to determine the identity, how much of these different RNA molecules are actually present, including the mRNA transcripts. Yeah. Now, what is this RNA sequencing? Why do you do that? By comparing these RNA sequences across different samples or different conditions. Maybe in one sample, it's a control sample, and in the next one, you're perturbing by a little bit, meaning you're changing something. That something can be adding some sort of a, drug to see how these cells actually respond, or how these conditions at the RNA level are changing, for example, right? So researchers can identify which genes are actually turned on. Again, I used another term called upregulated, downregulated. Okay, these are, again, in the parenthesis I put, basically it means turned on and turned off. And which genes are being turned on and off under certain specific conditions. And again, this is, and you do this on thousands and thousands of samples. And the collective information, you can gain insights, basically saying, okay, this is working, this is not working, and so on. So this is what the researchers are after. I took an excerpt from one of these publications from just a year ago, and I'll just read it out. It says, a comprehensive understanding of how these single cells, individual cells, employ their mRNA, right? How these cells are employing their mRNA and the proteins in different tissues of the human body. It leads to new strategies to prevent or treat infections. Basically what I just summarized. I mean, this is just an excerpt, and this can be very helpful in all types of research, like whether it is cancer, neuro, organ level. So it's applied throughout, the organisms research, not just a brain research like neurosciences, not just cancer, but this is generally useful for a lot of different types of research. Okay, now that we understand, okay, we need to do RNA sequencing or study this RNA sequence, what is sRNAseq? This is a term that you will again see quite often if you just started to get into this field. And also this is, like I said, an exploding field. So it's very good for you to understand what it is. SC stands for single cell, RNA is RNA, and seq stands for sequencing. So single cell RNA sequencing, which basically means you're getting information at a cell level rather than a collective level, a bulk level. So it permits the comparison of transcriptomes of individual cells. Now, I know I'm just reading off the screen, but you can read it too. But what is it useful for? Assessing transcriptonal similarities and differences within population of cells. So when you read these at a single cell level, basically it comes down to, you understand this, you have information down to a single cell level. Then by looking at this collection of these cells, you can say that, okay, something is happening in this part of the tissue compared to the other part of the tissue. Or these cells, or the RNA coming from these collection of cells is behaving in a different way compared to the ones coming from a different side of this part of this tissue, right? If you just do bulk analysis, you know what the bulk is doing, but you don't know exactly how that relates to the spatial. side of it, what part of the tissue is happening, where this function is affecting. So that's basically what scRNA-seq helps us with. I'm just again putting a workflow that I got from this publication. Again, excellent publication that you should definitely read. This is a general workflow of single-cell RNA sequencing. Basically, you extract your RNA transcriptome in a way, and once you have that, you convert that into cDNA, and cDNA amplification gets done. So all of this is for your sequencing. And at the end of it, what you would get is, as I mentioned earlier, a data set of each cell, where the cell is located, and for each cell, what genes are present in that specific cell, because you're reading this down to a cellular level. Okay, again, you get more information. This video is not about explaining this in depth, but basically providing an overview and then jumping into the analysis part. Although I'll explain a couple of things that I think are useful for us. Okay, this is, RNA-seq is good, but comes with some requirements, right? So how do you study at a single cell? That means you need to liberate these cells from this whole tissue and make sure that these cells don't burst. So all this, the DNA comes out of it, then it contaminates like this entire experiment, right? So you need to make sure that there is no stresses and there is no cell death and so on, which of course is possible. People are doing this scRNA-seq, but in spatial, in transcriptomics methods, the spatial information is preserved because we are studying an intact tissue in most cases. So that's the advantage of spatial transcriptomics. And again, there are many ways people do this, and the two common things that I am going to at least provide a high-level overview of is the ISH. I'll tell you exactly what that is in a second, and using the sequencing method like arrays. So these are the two common ones, I should say, even though ISS and microdissection is also something that people do. But let's have a quick look at these ISH and then sequencing methods, and then jump into our data. So of ISH, there are various techniques, like FISH is a common term that you would run into, which is basically fluorescence in situ hybridization, and Mirrorfish is one of the most common techniques, at least in the last few years. And Mir stands for multiplexed error robust fluorescence in situ hybridization, and every term has a specific meaning. But let's look at the high-level description of Mirrorfish. And this is right from the paper. Again, I have the link down here. I'm flashing too many screens. I'll make sure that I put all the references down as part of the description. So please don't worry about getting your hands onto these documents. Okay, now let's. This is a big summary, and again, there is a lot going on here. For example, we can spend some time talking about what Hamming distance is, right? So I'm not going to do that. I'm just going to focus on this first image where you get a clear idea of exactly how Mirrorfish actually works. So let's go ahead and do that. So this is nothing but a series of experiments. You have a single cell. By the way, this is a cell, and that's a nucleus, and you have a single cell, and within the cell you have all these RNAs, right? So these proteins down here, and this is experiment number 1, this is experiment number 2, this is experiment number 3, meaning you're imaging the same cell multiple times. But what are you doing between these images? You're actually, in fact, let me show you this. You're actually doing this repeated rounds of imaging, and image 1 has one fluorescence tag right there, and you kind of image this under a fluorescence microscope. And the fluorophores that light up tell you that, okay, these genes are turned on and some of these are not turned off. The ones where it's turned on, you'll see a fluorescent signal, and you kind of call that 1. This is like a computer. You have a 5-volt signal, it's 1. You don't have a signal, it's 0. So in the first round, it's 1. For the ones that are lighting up, and 0 for the ones that are not lighting up. Okay, now you image it again by adding more of these fluorophores, and then now you can actually call this sorry, I keep saying fluorophores. I'm blanking out what the term is. I'll add that later as part of the subset. As part of the text on the screen. But think of these as, okay, you are adding something that lights up. Okay, you're making them light up by adding in each of these experimental rounds. And in this round, you see 1 right there. It's lighting up. That's why the second term is 1. The second term is 1. Here, it's not lighting up. So the second term right there is 0. Whereas the same gene was lit up in the first round, so it was 1. So 1, 0 means in the first image, we get some signal. In the second image, we did not get some signal. And in the third image, you got a signal. And fourth, fifth, sixth, so you have the n digit. So you can encode this into 16-bit, for example. For 8-bit, you get 256 genes can be identified, 2 to the power of 8, right? If it's a 16-bit, you have 65,500 some genes identified, and that's enough. You have more, I mean, typically you have what, 30,000 genes or so. So 16-bit is more than enough to actually decode these numbers into specific genes. I hope that makes sense for you. Okay, so moving on to the next image from this paper. I just want to say, okay, now you have all these things lighting up in each experiment. So now you have, let's say, multiple experiments done, multiple imaging rounds, and now for each of these single cells, you have 1s and 0s, binary. And that's exactly what you see down here, right? So for each of these, you have 1s and 0s, and now you basically say, hey, decode them and cell segmentation. But once you decode, you know exactly what genes they are. And of course, you need to perform cell segmentation, meaning identify individual cells, segment them, separate them, so you know exactly where they are located, and you know exactly how to attribute these individual genes to that spatial location. The implementation can be very tricky, as you can see. At a high level, it makes sense. I hope it makes sense to you. Eventually, as I mentioned already a couple of times, what you get out of this entire experiment is cell number 1, cell number 2, cell number 3, where they are located, and what genes are actually found, and how much intensity from each of these genes. Because some of those genes you have very low intensity, you may as well ignore. Some are lighting up very high. Some are lighting up just because it's an error, because you need to do some error analysis here, because some may just light up, I don't know why, for some other reason, right? And you need to take care of those as part of your post-processing, but let's not get into that yet. This is the table that you end up with once you perform this analysis. Now, let's quickly look at the sequence-based. Again, there are various ways, various methods. I'm going to focus on one specific method by, what's the company name? 10x Genomics, and it's called Visium here. They also have a couple other product lines that do something slightly different, but the way these guys actually do it is you have some sort of a sample preparation right there. So on a slide, and they call it a Visium slide, obviously, and it contains a matrix of capture areas. I'll show you how the Visium slide, actually at least a graphic, how it looks like. By the way, this is taken right out of their web page, so I should credit them. And yeah, once you put the sample here, and each of these capture areas, they have unique barcodes, okay? And you place the tissue on that. So think of a sample where each area is barcoded, and you put your tissue sample on it, okay? And on these capture areas. Now, the tissue on the Visium slide is permeabilized, and it allows the capture. What that means is it allows the capture of the RNA molecules from wherever the tissue is touching these capture areas. And the RNA transcripts from these areas are bound to these capture areas now. So basically you have a tissue with this RNA, and you have like these barcoded, traceable, specific locations, and the RNA is transferred from the tissue to these locations. And basically you take that and you perform RNA sequencing using your traditional sequencing methods. And of course, there are certain stuff that actually goes on. First, you need to convert that RNA, reverse transcribe that into your complementary DNA or cDNA. This is the common step for any of these approaches, by the way. I didn't talk about this in the previous slide. But whenever you're doing sequencing, yeah, you are actually going through your RNA and then reverse transcription into cDNA and so on. In the previous approach, it was completely spatial based. So you're actually getting the information, both spatial information and the genetic information directly from from the sample itself, right? From how they are lighting up. And cDNA, why are you converting that? Because it allows for the amplification and then your analysis. And eventually what you need to do is this library generation step. Once the library generation is done, then of course now you start to do your spatial, your sequencing on this data set, on this library. And the sequencing, almost everyone uses the NGS next generation sequencing nowadays. Again, that by itself is such a breakthrough and such an amazing technology. It's also in a way imaging related, but you don't think of that as an imaging device. Anyway, that's a separate discussion, but this is typically the process using the Visium from 10x Genomics. And this is how the capture area looks like. Of course, have these coordinates, exactly where these are located, and you tag them using some sort of a barcoding. And one thing to note is these areas are a bit large compared to your cell size. So sometimes you are getting information from a single cell, sometimes you're getting information from 3, 4, 5 different cells. But spatially, you can identify exactly where these are coming from. Yeah. So that's the idea there. And of course, when you look at a real sample, so this is a tissue in the background, and you have these spatial locations overlaid. As you can see, within each of these capture areas, probably 3, 4, 5 different cells will fit in each of these areas. And now what you have is information about the gene at the specific location of your RNA at your specific location. So this is how the gene information is encoded, for example. In some cases, again, I have to look into exactly if Visium does this type of location information tagging along with the gene information, but it is customary for you to look at data sets that look like this, where the first part is the genetic information and the second part is the location information. So you can kind of trace them back to exactly what gene that is and exactly what location that is on this slide. But again, as I mentioned 3 to 4 times in the past in this video, eventually it comes down to what cell it comes from, what the locations are for that specific cell, and what genes are present in that cell. I call this spot and not a cell because each spot in this case is multiple cells. So, but in the previous case, you have individual cells and what the XY are. Okay, so that's it. That's basically our starting point. And with that starting point, I think we are ready to get into data processing. Everyone has their own formats and everything, but it's very easy, no matter who the manufacturer is, it's very easy for you to get your hands onto these data sets. And don't get me wrong, whoever the manufacturer is, for example, Visium, I know they offer a great data analysis toolkit, so you can actually use it, the tools to get the information that you want. But if you want to push the limits, meaning if you want to extract more information, that's not possible via the pre-configured software. I'm pretty sure most of you who are watching are coders, that's why you're watching it, or bioinformatics wannabes, or existing bioinformatics people, or researchers, I should say. With your own coding, you can, of course, extract any type of custom information. Okay, so as I mentioned, no matter what technique, I only mentioned about Visium. By the way, 10x also does, and others, they also do another method. It's basically, it's like drop sequence, for example. It's you have these beads with, again, barcoded, and they flow in some medium, let's say some sort of a fluid like water, for example, and then you have cells that are also flowing, and then they kind of merge together. And the idea is some of these cells get bounded to these beads, and they go into this oil, so creating some sort of an emulsion. So you have like this water in oil, so they form these bubbles, right? I mean, these droplets, let's say, and these droplets, the idea is in each droplet, you will have a bead and that is barcoded, and a cell that gets attached. So this is how you can also study at a single cellular level. But of course, in these cases, you'll also have some of the beads with no cells, some of the beads with multiple cells. So you need to do some correction. So the way you handle the data, the way you clean up is slightly different depending on what type of technique the data is coming from. But eventually, again, after the cleanup, you have something that looks like this. Okay, so no matter what the technique is, this is where we start. And the downstream analysis, what does it include? Of course, it includes quality control. It includes dimensionality reduction, because for each of these cells or spots, whatever you want to call them, you have 30,000 genes, let's say, or 20,000. In some, you may have 500. In some, you may have 200. It doesn't matter. But you have many, meaning you need to perform some sort of analysis. For example, principal component analysis, to bring all these dimensions down to like a handful of dimensions. So of all of these, what are the predominant ones, right? So you need to understand that. So you need to change these genes information into lower dimensional space. We'll go through that exercise in the next half an hour. And you need to perform clustering. Once you reduce the dimension, you can actually cluster all of these, but then trying to interpret that would be a nightmare. So that's why you do the dimensionality reduction and then do the clustering. In fact, if you reduce the dimensions to only 2, which we'll be doing, then you cluster on a 2D plot, then you can identify multiple clusters, and then you can do cluster analysis. Once you have multiple clusters, then you can do differential expression analysis, meaning which of these genes are differently expressing compared to the rest. So that type of analysis. And of course, once you do that, okay, now that I identify these, what type of cells are these? And go back to your map of the tissue and then attribute these to specific type of cells. And of course, you do spatial analysis, and visualization is always important because we are all humans. We tend to understand things better when we see, and that's where visualization comes into picture. And these analyses help. Of course, why are we doing all of these? Because you want to understand, get the biological insights and what's going on at a cellular composition, heterogeneity, and all that, spatial organization. Again, that's the whole point of this spatial transcriptomics. If you want bulk information, you can do bulk analysis, right? Spatial information because you want to map the behavior down to the tissue level. Okay, I think that's enough background information. Let's go ahead and jump into our Google Colab notebook. And before we start coding, I want to show a couple of papers. Let me go ahead and zoom in. This one, it says comparative analysis of Mirrorfish with bulk and single cell RNA sequencing. This is a good reading material if you are interested in understanding, okay, what's the benefits of Mirrorfish? What are the limitations? And what do other techniques do? This can be a good paper. And the other one is Spatial Omics Technologies at multimodal and single cell subcellular level. Again, this kind of provides a good explanation in case I fail to explain things in a way that you like to understand. Yeah. Okay, now let's go ahead and jump into the code. Here is the Google Colab notebook. Again, I'm going to share this. Look at the link under the description. I have already downloaded the dataset from this location, and in fact, it makes sense for us to have a quick look at the dataset. This is a Visium dataset. Again, they're nice enough or kind enough to actually make that publicly available. In fact, they have multiple datasets, so go ahead and play with it. And here you have Visium mouse CSV and raw data. Let's open this. Okay, so in this case, they have 5 samples, 1, 2, 3, 4, 5, and each of these, it's just a metadata for exactly what we have. And let's go into the raw data right here, and you can see all the 5. And let's open the first one. And you can see the summary here. In fact, I love this. I love the way they actually generate the HTML summary. You can see number of spots under the tissue. So you have all of these spots and how many spots are under the tissue. Yeah, you can actually get a nice overview there, and mean reads per spot and median genes per spot. How many genes are there? Median genes, per spot, about 4,500. That's pretty good. And a bit of metadata right there. I think they also have analysis. Let's click on the analysis part. So we will be doing some of this analysis today, actually. some part of this analysis. Okay, so that's that. And what else do they have? They have the H5 files, and these are the ones we're going to read, and these H5 files are pointing to these ones. Yeah, the raw matrix spatial. For example, if you open this, you get some CSV information about it. Go ahead and look at it, but primarily we will be looking at the filtered matrix that has the barcodes, the features, which means the genes, and the matrix itself, the matrix that I was talking about. The matrix is basically your cells and what genes are present in there. So these are the data sets, and each is a TSV file that's just compressed, and we'll be opening this. In fact, we are reading H5. Now that we understand the file structure for this specific data set, let's go ahead and open it. Now, for those of you who are working in R package, you have a different package to work with for this type of analysis. Again, I'm focusing on Python. I'm not an R expert. I'm a Python person, so the primary library that we use is going to be ScanPi. Some people say that, but let's go ahead and install the ScanPi library here. And this is, again, it can be used for visualization. This does 90% of what we want to do. And let's go down, and now let's go ahead and install and data. Now, I put a lot of text here. In fact, I took a time to actually put all of these, and you can see and data. If you want to look at what it does, it's a widely used data structure in the field of the single cell RNA sequencing, and it's part of the ScanPi library. And it's designed to organize. And data just stands for, did I put it anywhere? Annotated data. I probably have written that somewhere. But let's go ahead and install that. And now we will be, after this, we'll be loading the data. And we are going to use the Visium data from 10x, because obviously they made it easy for us to get our hands onto this data set. And it uses a spatially barcoded array. I already talked about it. It's called a Visium slide, and it's a blah, blah, blah. All that information I already talked to you about. So let's not worry too much about it. And the workflow of the Visium typically involves, I already talked about the workflow, of course, sample preparation, and the tissue can be fresh frozen, or it's a formalin fixed tissue. Spatial capture, again, you're loading that into their instrument, and you're preparing the library. Your sequencing data analysis is what we are trying to do right now. And we have our data set, and you have raw data directory. We looked at it, but we said, okay, let's look at the filtered directory. And the filtered data contains, obviously, the processed and filtered version of this sequencing data. There's a lot that needs to be cleaned up and all that, but the filtered data directory has it. And go ahead and read this stuff. Yeah, so this is for you to go through it later on. And let's go ahead and import our ScanPi and data pandas. Again, most of these are pandas data frame in the backend, so you need to perfect your pandas skills in general if you want to extend the capabilities of what ScanPi can actually do. And ScanPi actually does a lot, so you don't have to wrangle with data using pandas, but yeah, it's a good skill to have. What I mean is pandas. Okay, now that we have these, I placed this entire data set on my Google Drive, and I've already connected that. So all I'm doing is pointing this to the data directory, which is nothing but, I'm just looking at one data set right now. In fact, this one. In reality, you may want to load all of these and then perform individual statistics if you want, but then combine all of those. If it depends on how you devised your experiment, basically. Right now, we are only looking at a single one. And read the AND data. So now that this is just a folder path. Now we need to create an AND data object. And this is the object we are going to work with throughout this tutorial. So AND data object, let's go ahead and read Visium, and they have a few other reads right there depending on exactly how your data is structured. It can be H5, and that's what I'm reading right here. So let's go ahead and read it. Immediately it should give a message that, okay, hey, variable names are not unique or something. Yeah, variable names are not unique. To make them unique, call. By the way, variable in this case is gene. Basically, it comes down to gene. And it says, hey, go ahead and do this, make them unique. And that's the first thing we usually do. So make these unique. And now let's go ahead and print out and see exactly what AND data looks like. Well, what the metadata is. And AND data has number of observations, 2,987 in our data set. And number of observations is basically number of cells. Well, number of spots in our case. And 31,000 variables, which is 31,000 genes. Yeah. And it also has data frames for observations, variables, and UNS and OBSM. So the observations, like I said, are all the cells. So it has information about, is it in tissue or is it not in tissue? What is the row and column of that specific spot? So that gives you the information about the cell itself. Now for genes, it has gene IDs. It has the what type of feature, is it and genome. We'll look at those in a minute, but I'm just giving you a quick overview. Okay, all that explanation down here. So let's go ahead and start looking at the observation. And we should have 2,987 such observations, right? So you see how this is encoded right there, the spatial location. And is it in tissue? Yes. And where is it found? It's at 50 and 102 in terms of their coordinate system. Okay, now let's look at the variables. We're just getting familiar with exactly how the data looks like right now. Okay, and these are gene IDs, and there you go. That's the gene name, which is the index apparently in this case, and these are the gene IDs and so on. Now let's go ahead and start understanding, okay, exploring the data. What are the highest expressed genes? Okay, we have 31,000, right? What are the ones that are highest expressed? So all we are going to look at is highest expressed genes. It's very simple as that. You can also work with your data frame, but these guys have done an amazing job providing us more information than we need. You know, hey, the highest expressed genes are MT, which I guess which stands for mitochondria, and lowercase MT means it's a mouse sample. For humans, it would be uppercase MT. Okay, but this is a mitochondrial gene, and you can see this is the highest one that's actually expressed, and then the next one is CO1, CO2, and so on. So bulk of these are mitochondrial, and this is the one that's non-mitochondrial. Yeah. So we are just plotting the top 20. You can see where the mean, median, and all that stuff is right here. Okay, let's gain our understanding a bit more. First of all, in ANDData object, of course, there is another one that's called ANDData.x, not just observation, not just variables, but also X. This is basically the main data that's stored as a sparse matrix. A sparse matrix is because we have a lot of zeros in here. That's why it's stored as a sparse matrix. So if you want to look at that, go ahead and print it out. There you go. Yeah. So 2, 0, 0. This is the matrix itself of your cells and genes. Okay. Now let's go ahead and print all the genes. Again, we're still exploring the data. I'm asking questions. Okay. I want to print all the genes with non-zero expression in a given cell. So my cell may have 4,000 genes with non-zero. So I want to figure out what that is. And there you go. So these are all the genes. I mean, this is a huge printout. So you can see, okay, in the first cell, these are all the genes with non-zero expression. Okay. And I mean, I put a cell index of 20, so we are just looking at that specific cell. Okay, if I want to look at the first cell, what are the non-zero expressions in that cell? So you can see there's a lot of mitochondrial ones right there. Okay, enough with these numbers. Let's start to visualize some stuff so we understand what's going on here. So the PL it stands for plotting, and dot spatial, you're actually doing spatial plot, and we want to plot AND data. And data stores objects at multiple resolutions. So you have high res and you have low res. So I want to print the high res version of it. And alpha equals to 0.5 means set the transparency between the overlay and the underlying tissue to be about 0.5. So this is the image I showed you in my presentation. So you can see these are the dots, and in the background you have the tissue itself. If you want to only look at the tissue, change the alpha to 0, or you can just call this high res and color equals to none. That does exactly the same thing. So we can have a quick look at the tissue right there. Okay, you can see a bunch of nuclei right here, and imagine having a dot right there. Having a dot, you can see how 3 or so nuclei are under a single dot, which translates to 3 or more cells at that specific dot. Yeah, location, spatial location. Okay, now let's generate a spatial plot of cells using gene expression levels, right? So let's focus on the most abundant one, MTCO3. Where are they actually coming from? In addition to this, let's throw in a couple of genes like RORB, I saw the names before, and VIP. And first thing first, let's actually extract the gene names out of our var, and our gene names are indexed in our data frame. So let's go ahead and do that. Now, before we go ahead and plot, let's see if these are actually part of this gene names. And I know they are part of it. So let's go ahead and plot it. How do you do that? Again, pl.spatial, a data. Now color is RORB, VIP, and MTCO3. We're going to add these three plots. And let's go ahead and do that. And here you go. This gives a good idea of where these genes are lighting up. Well, lighting up is the wrong term in this case, but where are you seeing a lot of counts coming from these specific genes? So you can see that this RORB is coming from this specific area, even though you get some signal from throughout. And same with VIP, getting some of these. And mitochondrial, no wonder we get lots of counts because it's showing up a lot of counts around here and not many counts right around here. Okay, again, we are still in the phase of understanding the data. Now let's go ahead and start cleaning up the data and preparing the data. So typically, I would start by, anyone would start by computing the quality control metrics, because right now we don't have any quality control metrics. All we have is just the data itself. So we should go ahead and do that. But oftentimes you may be interested in some specific type of genes, for example, mitochondrial, or the ones that are coming from ribosomes and so on. So let's focus on the mitochondrial one. Okay, so what I want to do is with this and data object, I want to add another column that says, is this a mitochondrial gene or not? That's what we're trying to add. And this is where we are trying to, first of all, look at the ones where it's starting with MT. We have quite a few. I think we have 13 mitochondrial genes in our data right there. And we want to tag them as, yes, these are mitochondrial. And for all the others, no, these are not mitochondrial. So that's basically what we are doing right there. Okay, if it starts with MT, go ahead and add that true or false. So let's go ahead and print it. And now you see there is a new column called MT, and it says false because none of these are mitochondrial. And for the mitochondrial ones, it should say true. Okay, let's move on. Similarly, you can tag them as ribosomal genes. Download, I should change this. I typed so much text, it's bound to make some mistakes there. Okay, so you can tag them as ribosomal genes, and ribosomal are not tagged like mitochondrial, so they're not like MT. So you have to find a data set of all ribosomal genes, or a database, or whatever, a template. And here is the link where you can get a CSV file of all the ribosomal genes. And once you have that, let's go ahead and print it. It actually gives you a list of all of these. All we need to do is now go back to our data set, our data frame, and say, hey, do I have it? If so, for that specific one, go ahead and add a true to it. And that's exactly what, if it is in, go ahead and add this ribosomal, and it should say, yes, true or false. Yeah. We don't see any truths right here. So now we have two columns, mitochondrial and ribosomal. Now we are ready to extract some statistics from these. How do you do that? Let's first of all look at the entire A data right there. So you have, you see how for the variables, which is for the genes, we added this MT and ribo. Yeah, previously we didn't have that. We only had these three. Now let's go ahead and add a bit more by calculating the QA metrics and QC metrics, and it's very simple. All you need to do is process like the sc.pp.calculate QC metrics, and if you don't give any of these, it's going to do the metrics for all the genes that you have, but now it's going to, in addition, give you the metrics for mitochondrial and ribosomal, and in place equals to true obviously means we are modifying our A data object with this new information. You may want to assign that to something else if you want to keep the original A data object for some other reason, but I'm updating the existing one And let's go ahead and see. Now you see there's a lot more stuff we have here for observations for the cells itself. There is like how many genes by count, number of genes for each cell by count. This is something we need to know, right? What are the total counts that you have, and log 1p of number of genes by count. So it performed a lot of analysis. Same with the variables. Also, it added a few number of cells by counts, mean counts, and all that stuff. Now let's go ahead and use this information in the next step, which is get some basic information. Okay, how many spots do I have? Mean reads per spot, median genes per spot. It should typically match. Didn't we look at? So let's say number of spots under tissue 2987. Let's go. This is the summary given by 10x. Yeah, 2987. They should typically match, obviously, especially these type of metrics like mean reads per spot, median genes per spot. You can extract all that. Mean reads per spot and median genes per spot, 4,500. They are very similar, not exactly the same. We have to go back and see why. Okay, now let's go ahead and look at our variable right here. So again, variable is our gene data. So there you go, all the gene data. You can slide this all the way to the right to get more information about total counts. There are 823 counts, mean counts, and number of cells by counts. And keep an eye on these. Like for example, this gene, this is important. Let me slide back. This gene right there, 0 cells by counts. This one 2, this one 8. Like we need to filter this data to make sure we keep only the ones that make sense. Right? So that's what we will be doing later on. And now let's also look at the observations just in case. And also for observations, you will see that, okay, number of genes by counts, you have all of these. But if I go to, I wish I could show you everything in one screen, but you can see, I think, where is it? Where is it? I'm looking for, it's making it a bit challenging. Sorry, guys. Let's go back here. Is there, oh God, or is it up here? Oh yeah, sorry. I wanted to show you dropout. It should be in this table. I don't see a dropout. Oh, dropout, percent dropout by counts. You see you have 99.9%, 100%, 99%, 100%. This one is a good one, 48%. And dropout is basically, did I put a note down here? A higher percent dropout value of a cell suggests a higher proportion of genes with 0 counts. So usually they are not of good quality. We need to drop them out. So anyway, so with this information, let's go ahead and follow italicized text. I should clean up this document before I share, but let's go ahead and get a visual idea of what's going on. So here we are plotting total counts as a function. I mean, the histogram plot of total counts. You can see 250 and number of genes by counts. Very good distribution right there. Okay, again, good idea of what's going on. And we can actually look at these. Like, for example, you have like 30,000 total counts right there, and above you have very few of these. So maybe we should drop all the cells with less than certain amount of counts. Same thing with genes, maybe we should only take like some of these genes and drop some of these. So now we have enough information. Let's go ahead and first start by sorting these total counts so we can actually see number of genes by counts at that location, only 3 and 38 and so on. And we have, on the other hand, quite a lot of these genes by count. So again, getting an idea of what is going on with our data, and nothing beats a violin plot. I absolutely love this because it gives you a lot of information in one single plot. So let's go ahead and look at this. So number of genes by counts, we should just take probably up to 6,000 or so, 5,000 or so, and then lower end maybe down here. And a lot of outliers up there, so we can actually set a cutoff at 30,000, 35,000-ish for the total counts. And also the percent counts of mitochondrial genes, we can set a cutoff at about 20% right here. And the ribosomal genes, the data is so small right there, we don't see much happening right here. So for the next one, we are only the high quality ones, let's say, or we are filtering our data. So by adding the filter cells, minimum counts 5,000, maximum counts 35,000, mitochondrial less than 20, just for the sake of completeness, let's put percentage ribo as less than 2, and then we'll look at the violin plot again. It should reflect exactly what we did, right? We did cut off at these locations, so that's what we are seeing. Okay, so slowly we are decreasing the amount of data to only what we need. And now let's also remove the genes that are expressed in fewer cells. So if the genes are expressed in less than 10 cells, okay, let's go ahead and remove those because we don't really care about those. We want only the ones with higher. Again, this is just a number, arbitrary number that I set up there. Okay, now the data is cleaned up. Let's start the analysis. What are we going to do here? We are going to first perform normalization and then logarithmic transformation, and we'll look at the results so you understand why we are trying to do this. But first thing first, let's go ahead and again look at, we did quite a few things. Look at the data, okay, fine. Now let's go ahead and look at the genes with most counts. What are the genes with maximum amount of counts, and plot it before and after normalization. This is just a fun exercise so you can see exactly what's going on. So we know that MTCO3 is the one with most counts. We already found that out earlier. And then this is just nothing but actually plotting that on a histogram plot. So it goes from somewhere around 100 to 1,200 over here. Okay, fine. Let's go ahead and normalize the total. So the goal of normalization is to bring the range into a similar range so you can compare them. If the range of one gene is low, if the range of another gene is high and all that, then it makes it a bit challenging for you to compare them, right? So to bring all of these into a similar scale, we are trying to perform this normalization. So when you actually perform the normalize, the scale here should change. You see how it went from 150 to 450. It's a much narrower scale now because we normalized it. And believe me, it happens to all the genes. It did the normalization to the entire data that we have. The next thing, this is not enough. We need to perform a log transformation. So again, in some cases, you may have these going from 100 to 1,000. Basically 1 order of magnitude higher, right? So to bring that into a comparable range, we perform the log transformation, and that's exactly what we do. And when you plot it, now you should see the range to be even more tighter, like going from 5.2 to 6.2. In fact, I should have actually plotted this for multiple genes. Then you can actually see that, hey, now all of these genes are in a comparable range. So now we are ready to actually go ahead and do our differential analysis where is this gene different than the other gene. For you to be able to say that, they need to be in a similar range, and that's exactly what we have done in the last two steps. Okay, now let's do this exercise of, okay, which of these genes are highly variable? Because some of these genes are probably not variable, they are uniform. The variable genes are the ones that actually tell us a story about what's happening in that collection of these genes. So that's again, read this text. I added that for you guys because I'm going to share the notes. But highly variable genes is again sc.pp.highly variable genes, and we are going to use Seurat approach to actually find out these. Look at the documentation to find out what other approaches, but this is so common, I'm going to use that. And we are going to only look at the top 2,000 highly variable genes. And that's what we do right there. And once that's done, let's go ahead and look at I print this quite often just to get a good idea of how the flow is. And now let's go ahead and plot it, because looking at that is one thing, plotting is better, right? So of all the genes, these are the highly variable genes up here. And this is, I think this is one with normalization, and yeah, this one, it says there, not normalized, and this one is normalized. That's it. Okay, now that we have 2,000 genes that are highly variable, let's go ahead and perform the analysis. And the analysis, like I mentioned earlier, dimensionality reduction. So we have so many genes. Now let's decrease the dimensions. So first thing first, let's only get the ones that are highly variable, meaning the first 2,000 genes. And you can assign this to a different variable if you don't want to overwrite a data. In fact, I recommend that, but I'm not doing it here. Okay, how do you reduce the dimensions? There are various approaches, but principal component is one of the most common ones that's performed. In principal component analysis, you can actually remap your data into n number of principal components. In this example, like pp.pca, if you don't say anything, by default, it's going to do 30 principal components. These are the 30 major components, but you can actually do how many ever. It's up to you. Now, let's plot the contribution of each of these principal components, because when you do the principal components, you are getting these 30 principal components. Now, these are literally principal components. There is no other name for that. These are individual components. And as you can see, the top 10 actually give you the most information about the data. If you look at this, PC1 contains the maximum information. When you combine that with PC2, PC3, PC4, and I think up to about, where does it flatten out? About 20, you can describe the data by using the first 20 principal components. So let's take the number of principal components to be 20. That's the step right there. Now you have your top 20 components. So what's your dimensions now? 20. We need to bring it down further. Let's do that by using this UMAP. Again, this is Uniform Manifold Approximation Projection. This is, again, a different way of reducing your dimensions. I am not going to get into details how it's going to perform, but let's reduce the number of dimensions to 2. You can do 3 if you want to plot it in 3D and do 3D clustering. Let's go ahead and do 2 components. And now let's go ahead and plot it once. This step may be a bit slow depending on your data set, but let's go ahead and plot them. There you go. Now we start to see a plot that you have seen here, right, in their analysis. Let's go back. There you go. Something that looks like that, because we're plotting the same data. Of course, it looks very similar. Although we are pre-processing it in a slightly different way. Now, the goal is to colorize these by dividing them into various clusters. First thing first, let's actually see, sort these by number of counts or number of cells by counts. Okay, so this one you have 2,985 and so on. Why am I doing that? Let's go ahead and plot these for these specific genes, and as you can see, I'm just taking the top 3 ones, yeah, and you can see that, okay, for this specific gene, most of the counts are coming from this area. For this one, FDH1, most of the information, you have lots of counts coming from here and not as many over there, and these you're getting a lot of counts from here. Okay, so now that we have our 2 dimensions, now let's go ahead and perform cluster analysis. And for that, you can use k-means clustering. You can do various approaches, but let's go ahead and install a library called Leiden ALG, Leiden algorithm, I guess that's what that means, or Leiden algorithm, and it applies this Leiden algorithm basically. Yeah, so we're not doing k-means, but we're doing this Leiden approach. So let's go ahead and add that, and this resolution term is something that you have to play with. You perform this Leiden clustering, you go and look at how the clustering looks like. In fact, let's do that. And when we do perform this, all it's doing is, when you perform that analysis, let's take this all the way to the right hand side. You see how it added something called clusters, a column, and you have 1, you have 0, 3. It's basically the cluster number. It assigned each cluster a number, and it tells you what cluster number this came from. And let's go ahead and map it so it's visual now. Yeah, so there you go. So this is the clusters, and you have 9 such clusters. And if I go back and change this to 0.6, we will get 0.8. Let's do 0.6 for the fun of it, and let's go ahead and plot it. And now you see how we have 12 instead of 9. I'll just leave it to 12 for now. Yeah, so and then proceed. And see how, I don't think I have to change my code just because I have more clusters now, but we'll find out. Okay, we have 12 clusters, and each cluster spatially, when you map it back, has to go back to something that's meaningful. That's the whole point of this exercise, right? So let's go ahead and plot the total counts and number of genes by counts for now, and have a quick look at the plot, and then continue. So there you go. So when you plot this total counts and number of genes by counts, you're actually spatially looking at, okay, I get a lot more counts from here compared to this region, And same with number of genes. I see a lot more genes being expressed in this region compared to here. But we will also plot the one that we just did, which was clusters, on top of this tissue. So you can see exactly if these clusters are mapping to some unique areas. This is great. In fact, I'm glad I did this 12 clusters, because you can see this new one, this area, these collection of cells are expressing genes that are clustered right here. And this one, each of these clusters. Now we can actually go back and map this to, if you find out exactly what this is, what that type of tissue is, what these type of cells are, then you can go ahead and label them. So you can say, hey, these are skin cells, these are endothelial cells, or whatever the cell names are, you can go back and label them. Right now the labels are 0 through 12. That's it. Okay, so by the way, a quick note, you can actually, instead of working on the entire data set, you can actually taking a subset. So I'm going to leave this 2 lines of code here, so you know exactly how to extract a subset from this data set, but we're not going to do that. But the most important thing now is to find the marker genes. Yeah, and marker genes are, in case you don't know, they're known to be expressed in specific cell types or conditions. That's what we're trying to figure out anyway. They serve as the indicators, or these are the markers that help us identify and characterize these cell types. So let's go ahead and find these marker genes. What are the marker genes for these different cells? So first thing first, let's go ahead and rank the gene groups right here. And for that, we are going to use a t-test. Again, read the text right here. When we apply this, it performs differential gene expression. That's exactly what we are trying to do. Yeah, between the different clusters. How are these clusters different than the other cluster? These genes different than the other ones. So that's what we are trying to do. And it compares the gene expression profiles between these clusters. It's doing a lot just with one line of code right there. And for that, obviously, when you're comparing two collections of data, t-test can be very useful. That's what we are using to compare that. Okay, now let's go ahead and rank them. Now that we did it, and rank the top 10 of these genes, and you can actually see or plotting them, and you can see that, okay, there is the score. It's very tough for you to see right there, but you can see how in some cases you have these genes. In some cases they are kind of flat over there. But more importantly, let's actually do a heat map instead of doing that. And here, oftentimes you can actually see, okay, there is this NRGN, which is kind of showing some sort of a hotspot right around there, and some other genes showing some hotspot somewhere else. So let's actually pick a couple of these. NRGN is okay. Am I using NRGN? PPP. With the previous one, this is the one that I picked. But now that we changed, let's actually pick something else that could be of fun. Let's do, what is that? CTXN1. So let's go ahead and use that here, CTXN1, and let's go ahead and plot it on our tissue, so we can see exactly where that is being expressed, where that's actually showing up. So there you go. So on the left-hand side, this is our clusters, and where is our CTXN lighting up? It's actually lighting up all around here. Okay, play with this and change this. This is where you need to iteratively go back and create different number of clusters. Maybe you don't know what the optimal number of clusters is, right? So by looking at it, you kind of get an idea, but this is where you spend some of the time. You go back, change the number of clusters, and come back and look at exactly where what's happening, and then get a good understanding of what, how you can actually map these clusters to spatially here. Okay, we are approaching the end of it. Believe me, print the top n genes per cluster. This is something that you may be asking a question, right? So, okay, I have a cluster in cluster 1 or 0, for example. That's our first cluster, right? So what are the top genes? So these are all the top 10 genes in that cluster. And this gives you information in terms of what type of cells these are in that specific cluster by mapping this to some sort of a cell, information database. I think I put a link to, you can go to the protein atlas and go back and look them up, reverse look them up and say, oh, this is commonly found, that specific gene in these type of cells and so on. Based on that, you can come back and label them. Okay, I think I should stop there, and I just want to give you a quick idea or information about various other libraries. We just focused on ScanPi, but there is something called SquidPi, which is built on top of ScanPi, and it does a few additional stuff. Again, I started adding something from there, some code trying to explain from their help manual, but then I really find their help to be super amazing. So just go to SquidPy documentation and you'll get a lot of information about it. So I'm not going to bother you, talking about it. And you can also look at cell to location. This is another useful library for spatial mapping. So please go ahead and get more information on it. And then there is another library called SCVI. This is another amazing library that's relevant to this. I'll try to, based on the response that I get for this specific video, I'll try to delve into some of these, but I just want to make sure I give a quick overview to those of you who are interested in getting into this type of field or changing your biological, if you're a biologist, changing your focus, of course. I hope you found this long video to be useful. And if you are a data scientist trying to find some topic, that interests you, I hope this interests you. And again, thank you guys very much for your patience. Let's meet again in the next tutorial.